in biomedical engineering from St. Louis University and her PhD in bioengineering from Georgia Institute of Technology. Her current research projects include nanoparticle delivery after brain injury to neural injury biomarker discovery to neural tissue engineering regenerative medicine. Her research is funded by many federal grants, NIH, NSF, state ABRC, and private foundation clinical sources, Flint Foundation, Phoenix Children's Hospital, Mayo Clinic. She has received a number of awards, including NIH Director's New Innovation Award, NSF Career Award, Arizona Biomedical Research Center Early Stage Investigator Award. Additional honors include Emerging Investigator of Journal of Materials Chemistry B, Emerging Investigator Biomaterial Science, Fulton Exemplary Faculty, Emerging Investigator Journal of Biological Engineering, and the Society for Biomaterials Mid-Career Award. The floor is yours.
Okay, thank you. Make sure everybody can hear me. Okay. All right. I wanted to thank everybody, or thank the Institute for the invitation to speak today. I wanted to share today with you a little bit about the work that my lab does, and mainly the nanoparticle and biomarker discovery space. So we'll just kind of dive right in. Okay, so just a little bit of overview in terms of the motivation for my work that my lab does. So my group focuses on traumatic brain injury. I just want to highlight that this is a substantial health and socioeconomic problem in terms of the care that is required and the loss of work and due to deficits and cognitive impairment that patients may incur. And so we see over 1.7 million TBIs that are reported annually. A third of them are related to injury, contribute to the injury-related deaths. Again, substantial economic burden on the healthcare system. And then just have some statistics here about recent military incidences in terms of the recognition of TBI being not only a single event, but potentially could have chronic and prolonged implications on a patient's life. Okay, so just broadly, before we dive into talking about different ways to address and diagnose TBI, what is a traumatic brain injury? So a TBI occurs when a person sustains some sort of mechanical perturbation that is then propagated to their brain tissue. So this is called that primary injury. And so this could be from a fall, through a car accident, when you have some type of explosive device, some shock wave that may impart this mechanical perturbation to the brain. And so that can lead to immediate neuronal death, axonal dysfunction, and hypertension. And then that kicks off what's known as the secondary injury cascade. And so this is a little bit more complicated scenario where you may have microglial activation, neurotoxicity, astrogliosis, inflammation of peripheral inflammatory cells, breakdown of the blood-brain barrier, so dysregulation of that normal crosstalk between the cerebrovascular system and the brain, edema, hemorrhage, and oxidative stress. And so I guess what I wanted to impart, and then to show this, how this injury process plays out, is you actually could see that this occurs not only immediately after the brain injury, but this is also a long-term prognosis or a long-term effect on a patient's life. And so the injury cascade can initiates minutes to seconds after injury, but then you can have signaling and processes that occur weeks to months to years after injury, it's been shown. So when you start to think about this from how to approach this from a therapeutic or a diagnostic point of view, we need to think about the various phases and points after injury in which we want to target. So you can see this in terms of that healthy brain microenvironment, and then how this can change in terms of your acute microenvironment to the chronic microenvironment. And so basically, we're just looking at this as a very complex temporal and spatial pathology that can ensue and progress over years. So my group is, like I said, so in terms of what my group targets within this traumatic brain injury space, is we're interested in developing both diagnostics and therapeutics. And we're using that by a few different mechanisms. So first, we're doing biomarker discovery. So I'll touch on this towards the end of the talk, where we're using phage display to develop novel traumatic brain injury markers. We're also, a central pillar in our lab is using nanoparticle systems to look at them for diagnostic and therapeutic applications. And I'll talk about that, the work that we've done in that space here today. And then I also have another arm of the lab that looks at regenerative strategies. In the past, we've looked at endogenous neural stem cell signaling, so ways to potentially recruit and tune the inherent endogenous regenerative signaling that occurs in the brain, and then also more traditional neural tissue engineering approaches as well. I'm not going to cover those today, though. So focusing on that nanoparticle piece, my lab, when we came into this space over 10 years ago, about 12 years ago, there were very few publications in this space. And so now if you search TBI and neurotrauma and nanoparticles, you'll see a number of different folks that are entering into the space. And I think that's fantastic just to see how we can leverage our understanding of nanoparticle delivery and utilizing that as a potential therapeutic option. So it's been very exciting to be in this space. So when I started, there were three main areas that I identified for my group to pursue. So first is just understanding, I would say, the basics of nanoparticle delivery to the brain. So can we cross the blood-brain barrier? Is it more susceptible, more permeable after brain injury? So some of these questions had not been reported, and my group has provided some seminal work in terms of understanding how nanoparticles may move in the injured brain. We also look at nanoparticles for sustained and localized delivery. So what are the different characteristics and design parameters to maximize therapeutic efficacy? And so I'm going to share one piece of therapeutic material that we've been looking at. And then the other is molecular targeting. So if we start to decorate these nanoparticles with targeting domains that may stick and hone to the injury at different time points, does that afford us better options? So one thing is just being able to first design novel targeting strategies. And so I'll share the story of looking at some phage display for designing some new targeting motifs. So first, that first looking at just generally nanoparticle delivery to the brain. So I wanted to highlight this work was done by a previous student in the lab who is now has her own successful career and is currently, I think, a research scientist and has her own lab at Eli Lilly now. So Vimla, when she came into the lab, again, we kind of were just starting to look at this nanoparticle delivery. And so what we wanted to do is just characterize the blood-brain barrier disruption and nanoparticle accumulation in a commonly used focal TBI model in the mouse. So we used a cortical impact injury model. With those of you that are not familiar, this creates a focal injury, and you'll see the tissue and histology for that. So essentially there is a localized necrotic lesion cavity that forms, and you see very heightened pathology with that. So the parameters were moderate to severe. We did our CCI, and then at different time points after injury, 0, 21, 3 days, and 7 days, we injected a cocktail of 40 nanometer pegylated particles that were polystyrene. So these are flow spheres, just inherently fluorescent, so we could track them. And then also an HRP injections. That's basically our small molecule delivery system, just to corroborate blood-brain barrier disruption with nanoparticle areas of accumulation. Did some cryosectioning for HRP analysis and fluorescent microscopy. And so with that, this work we identified, and I'd say in terms of the CCI model, blood-brain barrier permeability is a well understood and previously explored space. And so we knew with that small molecule of HRP that we have what's traditionally known and seen in this CCI model as a transient blood-brain barrier disruption. So you have the highest blood-brain barrier permeability that occurs immediately after injury. and then decreases at 24 hours, and then increases at 3 days. This is thought to be more of a inflammatory response mediated contribution to that blood-brain barrier disruption at the 3 days, and then typically resolves by 7 days. And so this data had been published in the late 90s, and so we knew that basically that this was the result we were expecting. So we were just kind of demonstrating that we could replicate this data and show similar trends. So then when we start to look at the nanoparticle deposition, you can see here at the different time points, 3, 24 hours, 3 days, 7 days. Pink is the 40 nanometer particles, blue is a DAPI stain. And we can see very similar trends in terms of the area of the nanoparticles that accumulated. So significantly high immediately after injury, small amount at 24 hours, and then again, significant at 3 days. And so basically this kind of helps us dial in to where we would want to begin to deliver, and that basically nanoparticles could be feasibly delivered to the injured brain and at least accumulate in that injured area. And so one last study that Bimla wanted to do and we decided to pursue was to look at sex dependence, or actually just see if there was difference in the nanoparticle accumulation profile based on sex. And so what we saw was actually that, yes, we did see a significant difference in terms of this 1 day time point where the females saw a more graded slope in terms of, so they still had this sustained high level of blood-brain barrier disruption at 1 day that was comparable to 3 days compared to the male counterparts. And so this actually spurred a recent R1 that we have, and we're actively pursuing this space. I won't be sharing any of that data today, but it's a very exciting story that we're very intrigued to look at the potential for sex to contribute to variations in terms of blood-brain barriers that may contribute to pharmacokinetics and dynamics. So another study that Bimla did, this was the last study she did in the lab, was to look at the nanoparticle delivery. So one question we always got was, and we were just kind of wondering, all of those analyses that we'd done previously were all static snapshots via cryosection. And so we were interested in looking to see whether or not we had extravasation of the nanoparticles into the parenchyma. And so what we did here was we used 2-photon microscopy in collaboration with a colleague across the way, U of A College of Medicine, Phoenix, that had an intervital 2-photon microscopy. And so with this, we were able to use a cranial window setup, and we have our sham animals where you can see this nice outline of our blood vessels. Red in this image is 40 nanometer particles. Green are 500 nanometer particles, which we had tested previously in another study. And then you can see after injury, acutely after injury, 3 hours, we can see that we have stark, I would say broad extravasation of our red nanoparticles, our smaller nanoparticles into the brain parenchyma. Whereas when we had these larger 500 nanometer particles, I use the analogy of like a storm drain or a pipe that has some sort of sediment of different particulate size, and the pipe has different holes in it. So you can see that the 40 nanometer particles were able to extravasate and move across the pipe barrier, whereas the 500 are kind of getting stuck along those leaky holes. And so that was interesting to us to see that phenomenon, I would say, in real time. And we're able to then run these images. So these are all just intensity gradients that we acquired from live videos, but they're the still images representing the distribution of that. And so we're able to demonstrate that, yes, we were able to see this extravasation at our 3/24 3-day, and then it's pretty much resolved here in this study that we did here. Okay. So essentially with the kind of the story with that body of work was that we're able to establish parameters for us to look at nanoparticle delivery and start to build a foundation for us to move forward with nanoparticle delivery for TBI. So we know that we did have a sex-dependent delivery window. It seems that 1 to 3 hours, 24 hours, and 3 days in our moderate to severe injury model is appropriate for us to pursue in our focal TBI model. I would note that one of our papers, so this, I think the nanomedicine paper, the 2018 one, we actually looked at different types, so a diffuse injury model, as well as a repetitive closed head injury model, and looked at nanoparticle accumulation in those, and we were able to report differing profiles. So it is injury phenotype dependent in terms of your windows for nanoparticle delivery, just based on passive permeable aspects in terms of that are related to the TBI pathology. So we were excited about this and said, okay, let's start to think about what we can do further to develop some novel therapeutic TBI strategies. So I'm going to tell you a quick little story looking at nanoparticles for localized delivery and how we can deliver in a therapeutic that we were interested in. So in this story, this was work that was solely done wholeheartedly with collaboration with Dr. Rachel Sirianni at UMass Chan Medical School now. And so we started chatting about different nanoparticle systems and things that she may have on board to that she had in terms of looking at it for cancer applications. And so we were talking about the role of histone deacetylase in TBI, so HDAC. So HDAC had known that in terms of looking at as an epigenetic modulator, and had been known to, if you inhibit it, had evidence that it would maybe be an appropriate therapeutic for acute brain injury. And so you can see that kind of body of literature here. And from our knowledge and from our search at that time, we had not seen any nanoparticle work that had been done in that space. And so at the time, I had a very talented undergraduate student, Georgie Musso, who is now in his PhD at UC Berkeley in neuroscience. And so Georgie came on board and said, yeah, I'm interested in looking at this particle system for TBI. And so what does HDAC necessarily do, or an inhibitor? So we're looking at essentially the on and off and kind of the control of that chromatin remodeling, so that opening and closing of the histone or the chromatin for genes to be turned on or have more access for the reader to be turned on or off. And so this regulation, again, has been shown to be important and critical and altered after traumatic brain injury. So we had, with this system, Rachel's group had developed a polylactic polyethylene glycol, PLA-PEG nanoparticles that could then deliver a histone deacetylase inhibitor, an HDAC. So the specific one is cusinistat. And so this was interesting from the standpoint that it was known as the second generation of HDAC and inhibits both class 1 and class 2 HDACs. And so previously, Rachel's group had shown that they were able to achieve 9% drug loading, and that you could have slow release on the order of hours compared to free cusinistat. And then also in their model of glioblastoma, they saw a significant therapeutic efficacy when they had the cusinistat nanoparticles delivered in terms of tumor growth in their model. And so we are interested then to look at this for traumatic brain injury applications. So the first question we said that I'm going to share with you today is, does the administration of the cusinistat nanoparticles increase histone acetylation levels acutely after TBI? And so essentially, we're just looking at basic pharmacodynamics. So we're trying to identify whether or not a single dose of these cusinistat nanoparticles would have an effect on that cortical histone acetylation levels. And so we're able to then look at the relationship between a dosing and whether or not we're seeing this activity in effect. So we had our CCI model, an hour. So again, using that knowledge from our prior nanoparticle foundational study, inject these nanoparticles retroorbitally at 3 different dosing regimens. And then 6 hours after that, we then collected the cortical tissue and then dissected it into the injury site, the ipsilateral cortex or the contralateral cortex, remaining ipsilateral tissue. And so we did some tissue lysates, some Western blots, and we're able to quantify and demonstrate that we're seeing a dose dependent response. And so we saw only significance compared to the blank nanoparticles in terms of the acetylated H3 versus the total H3 that had a significant impact. And we saw that was localized to that injury region within the cortex, and was not a broad distribution there that we saw a significant effect. So we moved forward and said, okay, does this have any functional, alleviate any injury-induced functional deficits? And so we had a little bit more complicated testing paradigm where we had our CCI model, injected our nanoparticles retroorbitally, only tested that high quisinostat nanoparticle dose. And again, what I want to emphasize here is we only did one single injection of the quisinostat nanoparticles. Other studies that had used HDAC inhibitors typically would do daily dosing for weeks. So we designed this just to kind of see a single treatment would we have an effect. So what we did here is we did some pre-training of our animals on open field, as well as grid walk, just to kind of get a baseline in terms of their locomotion and fine motor skills there. And then we did the CCI, injected the nanoparticles, and then we then did a battery of tests for over a week or over a month, so 35 days. So we tested rotorod for the motor skills, open field for locomotion, grid walk for fine motor skills. We also did a spatial learning Morse water maze analysis at the end of the study. I'm not going to cover that today, just out of time's sake, but we did have that assessment as well. So basically what we're looking at here is we are demonstrating that the quisinostat treatment, we saw that it dampened motor effects or motor deficits as identified through our rotorod. So essentially the mice are placed on a rod that is accelerating to 400 to 400 RPMs over 5 minutes. And you can see first here that our sham animals, so these animals received the surgery but not the injury. You can start to see this deficit emerge in our injury animals at 3 days on this run, and was sustained out to 7 days. And then when we start to overlay our blank nanoparticles, BNP, so that's an orange, and then our quisinostat nanoparticles here, you can see by day 7, we're starting to see that a dampened motor effect. So this was significant compared to these other saline groups, and also the sham group. So that was, again, encouraging to us to show that we're able to dampen that motor effect, and from there. And then the next one we looked at is grid walk. So again, just showing you first that we were able to detect a injury-related deficit at day 1 here, with significance, and these are ends of 11 to 16. So we had fairly large groups. You can see the variability that's just inherent into the animal data. And then we can start to see that we have the quisinostat nanoparticles had significantly fewer foot faults compared to our injured with saline, and that it was out to 21 days, we started to see that significance in terms of the number of footfalls was also low there. So again, this is encouraging to us to show that therapeutic effect. And then the quisinostat in terms of how that may affect open field. So open field at this for this study, mice are placed in an open plexiglass clear chamber. A light is placed at the top along with the camera, and then you look at their movement within that space. And so you typically have it's a test of general locomotion, but also you can start to get at some of their anxiety effects in terms of how long they're spending or how much time they're crossing an open space, because they typically like to stay towards the edges for just kind of inherent safety and predator avoidance behavior. And so when we're looking at this, we can start again. So with this, we saw only at the 7 days that there was a difference in terms of the distance traveled, in which we saw that the injured animals actually had a slightly higher locomotion activity. And then we saw that we actually had a decreased level of that activity in terms of locomotion for the quisinostat-based nanoparticles that was closer to the sham or baseline. So, and then at the end here, we're just showing a little bit of RNA-seq data where we're starting to see the emergence of a reduction of chronic inflammatory mediators that typically emerge after CCI, so the saline group, and then also return of baseline microglial homeostatic markers with our quisinostat treatment. So we're very excited about this and kind of looking at this in terms of positioning us to potentially move forward with other HDAC inhibitor treatments for TBI and seeing that dampened motor impairments. We still have a lot of questions in terms of dosing, timing, and just kind of additional molecular mechanisms that we could be probing there. So, but this is a first good initial study for us to come through with. Then the last story I want to tell today, just looking at the time, I think we have enough time, is looking at designing unique targeting motifs for TBI. And so in this space, so when you begin to look at biomarkers to, well, diagnose TBI, you can start to see the complexity of that mirrors, again, that evolving temporal pathology that I showed at the beginning of the talk, where you can have kind of bin these into like acute biomarkers, subacute biomarkers, chronic biomarkers, and you can see variations in terms of the different relationship of your biomarker to that time point after injury. So relation to gliosis, neuroinflammation, neurodegeneration towards this chronic time point here. And so I would say the TBI community has been pursuing this space in terms of looking at, I would say, mainly liquid-based biomarkers, so blood, CSF based biomarkers. And so Banyan Biomarker has developed the first FDA-approved brain trauma indicator biomarker, and that was approved in 2018. Based on UCHL1 and GFAP. And this is now used in the clinic. From my understanding, their specifications is to rule out the need for a CT scan among patients that come in with a potential TBI in the emergency department. And so that's, again, it's not a long-term, do I have a concussion? Am I ready to return to play? Looking at prognosis, it's basically whether or not a person may need a CT scan. So whether there's evidence of disruption of the blood-brain barrier and hemorrhaging. And so I think there's a lot. So this is a great first step, but there's a lot of improvement we can make here. The other thing to note here is these are all based on liquid biomarkers. And so if we're looking at being able to target a nanoparticle and stick to an area of injury, these may not be the most appropriate targets to go after. And so I had a student, this is Dr. Brianna Martinez. So she graduated from the group, and her project was basically saying, could we develop, and so looking at targeting strategies that would exploit the unique molecular alterations that are associated with neural injury pathology. So will that enable an increased sensitivity for targeted diagnostics and therapeutics? So again, we're trying to look at what is changing within that brain microenvironment, and could we leverage that change in order to develop more sensitive ways to target TBI. So we do this via phage display. So if you're not familiar with phage, it's a large screening tool that is based on bacteriophage in terms of being able to do genetic manipulations to present a biological motif on the surface. So this is an example of displaying antibody fragments on the surface. You can also display peptides, DNA sequences. There's been a number of different innovations in this space. And so I like this because you can then have 10 to the 6th to 10 to the 11th. I think our library has 10 to the 10th or 11th, diversity of different ligands that can be presented and start as a screening, combinatorial screening tool. And so it's routinely used. And so my group is basically interested in using a DAB, so a domain antibody fragment that's displayed on the surface. So this is only the variable heavy chain of an antibody. So this region here. It's fully functional. It's based on a human framework. We have randomization of the amino acid sequences only within the complementary determining regions, CDR regions 1, 2, and 3. And we'll get more into that detail here in a minute. So my group is very interested, and we've used this library extensively throughout the years. So essentially, with the way we set this up is you can then start with your phage library, grow up, purify your phage, and then have your phage or your target of interest that you're interested in. The phage that have high that bind to your target, you bind those, elute the ones that don't, and then use trypsin to cleave and collect your phage, and then use those phage to then infect your new batch of bacteria to then grow up and have a directed evolution of phage population that are displaying motifs that have high affinity to your target of interest, in theory. So my group decided to do, or what Brianna did, was she took this and was then able to, she was able to then take this, and we ran our screens actually in vivo. And so what we did is we took our stock phage, we then did a controlled cortical impact, and at different time points. So we targeted 1 day after injury, 7 days after injury, 21 days, and then we also had a control sham injury study that we did there. So we did 2 rounds. So we essentially took our phage library, did our CCI at the different time points after injury. We then injected the phage retroorbitally, so they were in circulation, allowed them to bind to the brain tissue, extract out that brain tissue, extract the phage, and then have an enriched library for the next round of analysis. So we only did this twice. And then we use next-gen sequencing, so the power of bioinformatics for us to be able to use that. So if you're used to phage display, I would say this is a shorter number of rounds, but we were able to lean on some prior literature that showed doing next-gen sequencing analysis provides a robust pool or population analysis such that you don't necessarily have to do additional screens. And so we're thinking about that in terms of a reduction of the number of animals we had to use. So with this study, so what I'm going to show you some data here in terms of the number of phage. So the question here was, were we beginning to get have evidence of a enriched library or enriched population throughout our in vivo screens? And so when you can see here with our sham animals, when we did comparing round 1, so the percent of colony forming units with phage that were from the tissue lysates that we extracted, you can see that there was not much of a significant change between round 1 to round 2. But as we're starting to see in terms of that ipsilateral, so remember we collected this phage population from round 1, and then enriched that, and then injected that again. So we're starting to see an increase in terms of the number of phage at the acute. Same thing with subacute. And I'm not going to show the chronic time point, just again for time's sake. So again, we're able to recover the phage from the brain tissue, and we're getting evidence that we're having enrichment of injury-bound phage within our culture. So again, I mentioned that we used next-gen sequencing in terms of being able to then looking at down selecting and identifying sequences and motifs of interest. And so typically what is done with phage is you use Sanger sequencing. So typically you spread out the colonies, pick single colonies, and then sequence those, and then look at kind of the sequences that emerge the most number of times. And so we actually decided at this point, when we started this study, there was beginning a shift in particularly with this longer DAB fragments and longer amino acid populations to use next-gen sequencing. And so we're able to do that because we could then have evaluate the entire sequence, look at the entire population, and then get the full coverage. So it seems like a great resource for us to pursue and use to be able to sample not just a small portion of our phage that were eluted, but the entire population. So another thing, just providing a little bit of terminology. So if you recall, I said that we have a variable a domain antibody library phage. And so we actually are presenting the variable heavy chain, which has these complementary determining regions. And so the variability of our library and the diversity arises from randomized amino acids in the CDR1, 2, and 3 regions. And so with that, you can see that kind of structured, highlighted here. So the CDR2 and 3, or 1 and 2 regions, are typically known to be a little bit more structural support, and that actually the CDR3 region is the most diverse and is the one that really determines antigen binding and specificity to epitope regions. And so we really focus the next bulk of our analysis all on looking at the CDR3 region and comparing those across within our phage samples. So when we do that analysis of our CDR3 regions, you can see that we were able to, within our libraries and kind of different populations from that second round, you can see that we're getting hundreds of thousands of recovered CDR3 from our libraries. But an important thing when you start to look at these Venn diagrams is that we have, I would say, largely or little sharing between the different conditions that we ran our screens under. So there's some convergence and some shared points, but the majority of the recovery sequences are unique to those distinct time points. So again, we're pretty excited about that finding. But then again, so at the beginning, I said our goal was for us to find and identify the motifs that would then bind to TBI at different time points after injury. And so you can imagine, so we're like, okay, we have this library now, we have 660,000 different sequences for us to think through. So then what we said is we're going to start to go through this criteria to select HCDR3s. So we wanted to make sure that, one, they're enriched through the biopanning process. They're unique to that time point, so they're not present in any of the other ones. They're also not present, and we collected tissue from spleen, heart, and then also had a internal propagation control. So we did a lot of kind of controlling and checking for those different to make sure that they weren't dominant in different organs as well. And so we could see that in terms of whether or not they were unique to the conditions, we could see that the libraries, they seem to be pretty much, so when we had started out with our first round with the acute, we had 60% that was within our control library, but then it dropped significantly down to less than 10%. And less than 15% for that second round. So that was good, again, to show that we didn't have that overlap there. And then when we're looking at that enrichment, what we're looking for in terms of that was to have a higher frequency of expression in round 2 than in round 1. So we just used some maps in terms of the reads per million that and to see that they appeared in our population, and the ones in the points in red, which it seems like it's dominating that, but we had those are the points that had that cutoff, so kind of cutting it off at that doubling for that frequency. And so less than 6% or 3% of our acute and subacute sequences were enriched respectively. And then we also then wanted to look at, again, that uniqueness to the specific time point. And so we utilized Z-score in terms of their relative expression in the different libraries. And so these are the top 20 highest frequency CDR3s in each time point. And then these columns kind of look at the comparison of whether or not they emerged or were present in the different time points. And so we focused on, so the ones that we kind of circled were the sequences that we then focused on for further analysis. And so kind of putting this together, a graph that shows the Z-score and enrichment values here, we can look at the relative expression of the sequences from the library to determine a threshold. And so again, kind of placing those priority scores in terms of the high Z-score and then high enrichment value, you can start to see that we're emerging and down to just a handful of sequences that we are really interested in looking at and pursuing further. So to kind of boil this down and summarize this selection criteria and what we started with, so we can see that we have a high number in the hundreds of thousands of recovered sequences. We have, we start to look at the enriched, so a smaller population, but still on the order of thousands. And then when we do that specificity threshold in terms of looking at that specific time point, now we're down to 500, but still in the thousands for the subacute. Those that were not observed in the controls were still in the hundreds to thousands. So what we decided to do there was we selected and started to test sequences with the highest fold enrichment value and the highest relative frequency. And so we'll call those A1 and A2 and SA1 and SA2. And I'm going to show you data just from those in yellow. And so again, just wanted to highlight that basically we had this wealth of data and wealth of information, and we're still utilizing and pulling from this rich data source that we have. But it allows us to, we had to have a stringent selection criteria to move forward in terms of what we wanted to test. And so when we're looking at like the A2, so when what we did is we were testing and utilizing and making some peptide mimetics that were specific to the HCDR3s. So when we made this A2 motif, we saw that it was binding and we had a high level of specificity to 1 day unilateral CCI. So again, that this A2 was that acute motif that we screen for in that system. And so then when we stain for this on our sham tissue, we do not see any positive staining. And so then when we went through, we characterized this in terms of the specificity for that temporal binding. We could see that our A2, so our acute, was showing a high level of specificity and binding on tissues that were acquired at that 1 day acute time point, low levels at subacute and chronic time points. But then for our SA1, so this is the phage population that was geared towards that subacute time point, we're seeing a preferential binding and staining towards the SA1, and less so, but still some at the acute and chronic. So again, this was interesting and exciting for us to find, have these findings where we're able to use the phage display to discover novel TBI target motifs that are temporally and are spatial and temporally specific. And so we're very excited about moving these forward and utilizing these in combination with our nanoparticle systems. With that, so I'll go ahead and end here. I think acknowledge the work that has been done by my group, and this is a really old group, but we have a lot of fun in the lab, and highlight again Brianna, Vimla, and Georgie that did a large amount of that work, and then no work can be done without awesome collaborators just across the valley here, as well as across the nation. Funding sources, and then I also want to put a plug in for recruiting. And if you think this work is exciting, I would say my work is just kind of the tip of the iceberg in terms of what you can do here at ASU. And so we're looking at recruiting awesome, excellent students to join our PhD program. And so I want to highlight and just kind of end and have this up here in terms of a QR code where you can learn more about our program, our biomedical PhD program, and reach out to our academic advisors to learn about application fee waivers. So with that, I will take any questions.
Thank you for this fantastic talk, Professor, about TBI. How do you get these biomarkers? How do you validate these biomarkers?
Yeah, so the phage, the biomarkers that we identified at the end there. Is that?
Question came at 1:30, so just before you started the final diagnostic section.
Oh, okay. So I think, so maybe they're talking about the graph that I showed in terms of what has been identified in the literature. So that was done by, I would say, more traditional proteomic analysis in terms of analyzing, doing mass spec, and various proteomic work, and comparing, a healthy blood plasma sample versus someone after injury. And so there's been a lot of work in that space. And I highlighted some reviews that have, I would say, done those justice. So yeah, that's mainly how that work has been done. We basically were taking a different approach from not knowing and not comparing and relying on, like I said, more of the phage, the power of phage to potentially identify and have an unbiased ability to kind of sort through changes in the tissue with our in vivo screening.
Yeah, I guess the slide was biomarkers to diagnosis of TBI. You have this big slide with gliosis, axonal injury, apoptosis. I think that, yeah. Another question is, how do you choose which nanoparticle to deliver the drug? Do you think PLA-PEG is the best nanoparticle for delivery of the drug to brain injury site?
Yeah, no, I don't, not necessarily. And my group, actually, we explore and look at many different types of drugs and nanoparticle systems. I think it's largely dependent upon the drug, so just the physical chemical properties of the drug and how it can be encapsulated. And then I think also it depends on, your delivery and release kinetics that you want to achieve. And so I think in some cases, a polyester-based system is useful. I think in other cases, if you want a short burst of release of something, then maybe some lipid particle that had some sort of trigger to have more of a burst release kinetic might be more appropriate. So yeah, so I think I would say I'm particle agnostic in terms of it just really depends on what you want to deliver, when, and how long.
Thank you. A blood-brain barrier is a difficult barrier to cross. Are there open-ended questions for coating nanoparticles that can facilitate crossing this blood-brain barrier?
Yeah, no, and I would say, I mean, this is also a very long-standing area and something I didn't touch on at all. So like I said, right now, all the work and the main focus of the studies in my lab thus far have been on exploiting just inherent pathology. So we know that there's disruption in the brain and we don't have to use any active mechanism for transcytosis of particles to get across the brain. And so I would say that, yes, that would be a next stage. And if you look at, particularly like our phage, that were the staining of those targets, I think they could potentially be more intravascular targets that we're seeing, and where we may be able to stain and localize particles to the vasculature or the injured vasculature, and then we would need to utilize some sort of active transport mechanism to have the particles, if that's what we wanted to do, to have the particles transcytose into the brain. And so yeah, so I think it's a complicated system. It really depends on what time point after injury, what type of injury, and again, what drug you're potentially trying to get across the brain too.
Thank you. We had also people at the Institute working on nanoparticle delivery to the brain. And we also have a group in organs on a chip. But most of the research done with that group, they did not bother organs on a chip. They tested their particles, and went to the small animals. Organs on a chip is an emerging technology. When do you think the organs on a chip may be ready or useful to complement the studies? People are taking nanoparticles, going to rats? When do you think a barrier would happen that people utilize organs on a chip more, or are they doing that?
Yeah, I think that's a great question. So from my knowledge of that space, I would say modeling, to me, the limiting factor is readily modeling the complexity of the blood-brain barrier. So as we know, so we have endothelial cells, we have astrocytes with end feet that are there, we have periocytes, we have a number of different cell types that give rise to the complexity as well as dynamic control of the blood-brain barrier. And so I think, to me, I think capturing that in vitro has been difficult to do. And so that's my understanding of those systems. And so I think, again, yeah, the science is going to evolve. I think we're going to get better models, better ways to recapitulate that system in vitro. I do most of my work in vivo. So I know, for example, in terms of like this trauma pathology, and you could maybe model this on, organ on a chip, but you have a mechanical perturbation, but then you also have the crosstalk between the immune system and the cerebrovascular system. And so then you have how does the inflammatory modulators influence the blood-brain barrier disruption? So I think it's thinking about kind of the intersection of all of those systems. So I think you can start to give some basic questions once we have, I would say, the science emerges in terms of developing a better model for that blood-brain barrier.
Thank you. Is retroorbital injection a better way for delivering nanoparticles to brain tissue?
No, so I should have clarified. So retroorbital injection is it's an intravenous route of delivery. And so that actually it's systemic. So in mice, we find that we have a better success rate doing retroorbital injections and tail vein injections. So particularly with the mice that we use, they're black sick. So sometimes it's hard to see the tail vein. So we can more readily have a higher injection rate than with tail vein.
Thank you. One final question. TBI in the site can be from an explosion, other things. The TBI you create using mouse. It could be in a nerve, tissue, blood vessel, skull, etc. Do you do fMRI type of study to understand where the injury is, or which part of the brain is the injury at?
Yeah, so I wouldn't say, so we don't use, I have and do collaborate with MRI experts. And so we have used MRI as an output metric. But I would say in our injury model on our mice, we have consistent, we have a specific procedure that we use. So typically the injuries are in comparable regions. There will be some variability animal to animal, but overall we have, I would say, kind of very reproducible injury regions and phenotypes that we produce. So I mean, you could do other injury models and utilize MRI. And like I said, we do that in some of our studies, but not all of them. It depends on what we're looking at.
Thank you. Well, that's all the questions I have. Thank you for your time, Professor.
Okay. All right. Thank you for having me.